Again, my name is Rob Schuler, and in this session, I'm going to be talking about how, in greater detail, how we organize, how you navigate, and some of the visualization capabilities specific to face-based data sets. So my focus is really on the data browser part of the overall picture of what Chris just shared. So just to give you kind of an outline of what I will cover, first, I'm going to present a few slides on the structure of FaceBase. I think it's good to have an overall idea of how data are organized, the key concepts, how things are related, because really our data browser gives you a way to navigate through that structure. So understanding a bit about the structure will help you when you get to the data to understand both what you're looking at and how to drill down and find, the specific parts of a data set that you're really interested in. Then I'm going to show you the overall layout of a typical data set display, just because how that information from the structure of the database gets laid out for you to view it and just see the different parts and find greater detail in it. I will also show you just a little clip on the face-based data citation, which Chris showed you some of our instructions on the website. Those have a really good reference to Nature publications about guidelines for how you should be citing data. So I'll show you what we do to help you have information to be able to make those citations in your works. And then I'll do a couple live demonstrations from our site. One, an RNA-seq data set, which has a built-in genome browser on the page, and then a second, which is an imaging data set, and this one has an orthogonal slice viewer integrated into it. So you kind of get an idea how different types of data, because FaceBase is a diverse collection of data, but how they're laid out in a similar way, and then some of the capabilities that show up depending on the specialized form of that data. So first of all, how data are organized in FaceBase, we have a diagram here. This is a very simplified rendering of our overall database structure, but these are really the key concepts and relationships that help you to get around it. So you don't need to know the whole database, all of the technical details of how data are organized in FaceBase to really get around and find information of interest to you. So this kind of serves as a sort of a high-level schematic, if you will. And I just wanted to mention that our design was informed initially by some of the minimal information models. And you may or may not be familiar with these, but over the past couple decades, a lot of work has gone on in the bioinformatics side of NIH and other related biomedical research about how we organize and share data so that we're sharing and collecting enough of it so that it's useful for downstream usage. So there have been models like MIBI, which was a general purpose biomedical investigation model in Miami, which was for microarray data some years ago. And then there have been more recent attempts, not quite standardized, but for a minimal seq data specification. So we think of how we organize data in FaceBase as being guided by the idea of a de facto minimal information model, just enough to be able to help you find the data and then, if possible, reuse it to the extent possible without having to drill down into greater details. But those are possible, which we'll see. Possible to drill down into greater detail. All right, so starting from the top here on the project. So the data in FaceBase are all contributed by initially members of the consortium and now really any members of the research community. And these can be individual labs, they can be larger collaborations, generally conducting hypothesis-driven research. I put a little asterisk there because some of those are funded specifically to generate data for sharing purposes in support of other hypothesis-driven research. So that is where the data in FaceBase, that is where it comes from. So as Chris mentioned in previous rounds of FaceBase, FaceBase 1, FaceBase 2, we had a hub and spoke model. So the spoke projects were really specifically funded to generate data that could be shared. It was always, though, guided by some hypothesis-driven research question. So that is the starting point here. It's useful to see what projects have contributed, because oftentimes if you find data in FaceBase, you might walk yourself back to the project and see other information that is relevant to you and also of interest to your research. Then moving down, we have the data set. Now, this is a collection of metadata and data files. It's intended to be a cohesive unit of output from a project. That is kind of loosely defined about what scale and what scope of this data set collection should be. It could be an output related to one set of publishable results, something that is a companion to a paper. That's a really good rule of thumb, but it doesn't necessarily have to be submitted in that way, and it may make sense to organize it. You might find data sets that have just a few files that were specifically related to another primary set of data. They were secondary analysis, or in other cases, there was an investigation that had an ongoing research activity that just that made sense to keep it all together in one large data set. So it varies, and the previous iterations of the FaceBase Consortium didn't really feel that it was appropriate to be overly restrictive. So you're going to find data sets of different sizes, but the intention is that it should be some cohesive unit of output that made sense for that project to submit as a whole. I put another little asterisk here next to metadata. You're probably all familiar with that term, but I do want to stop there, not assume that. And this just means data that describes other data. So you think about this in the case of like consumer photography. You take a picture, and in that picture would be baked in maybe the geolocation, where you took that photo. You could have the type of camera that took the photo. You might have the focal ratio, or so-called f-stop, kind of information like that. And in the terms of scientific data, there can be some of that type of metadata baked into a file. You can think about the resolution of an image, or perhaps the instrument that took an image, or the output of microscopy or something. But that's not always the case. And also there's a lot of pertinent information that fall outside of what is typically found in a data file. So this is where the rest of this structure comes in, where we want to collect information on experiment, biosamples, and protocol. So moving from here, we, of course, over to the left, we have the protocol. I mean, clearly we know, and you're all familiar with what this should represent, the methods and materials used to reproduce or produce your results. This, I just want to mention, though, it has varied a lot over FaceBase, and it's something that, in earlier years, how much information should be repeated in FaceBase data sets was kind of a big open question. A lot of times there are companion publications that really describe more of the protocols used to produce the data. So I'm just going to say it varies a bit. In this current iteration, we are working to provide, I would say, more rigorous guidelines. We are looking at things like the Nature Protocol Exchange. So they have a nice site up that kind of says what you should put in protocols, and they even have a place where you can deposit them if you want to format it according to the Nature publication guidelines. There are other resources that have shown up, like Protocols.io, which has kind of a nice online service. I'm not sure what exactly their business model is, but they have a nice interface for sharing protocol information. And in FaceBase, you can link off to a protocol, or for new submissions. But we're going to be putting more emphasis on this moving forward, and so more of that information will show up. But past data, I will just say we have some information relevant to protocol, but we didn't have rigorous guidelines in the past. And then, of course, you have an experiment, a particular activity or procedure that's carried out within the overall investigation. In this case, we often think of this as a more more fine-grained unit of that overall investigation. So I know these terms are all kind of synonymous, but we use them here in a similar way used in some other repositories where an experiment is kind of used synonymously with like a particular type and a focused assay. So, you know, it could be a particular RNA sequencing assay or an imaging assay. And it's related then, as we move down in this picture, to the replicates and the biosamples. So we don't run any like a tissue bank or anything like that at FaceBase, but clearly we do want to have metadata, the information describing this part of the experiment, so that you understand when you get to the data what it came from, what was under investigation. So these can be biological specimens, whole subjects, or other material used in experiment. In some cases, we're talking, we have some special fields for like cell characterization and cell line information. These are typically then organized in groups of, say, the control and mutant replicates. And then from that, moving down, then we get to the data. And this, you know, we have both some of that machine kind of generated metadata as well as links to the actual files. So, and these can be the original generated or perhaps derived results. Now, the benefit when we get here is that we know where each file is in the overall picture of scientific metadata. So they're all generally related to a replicate, which gives us the relationships to the biological material and the particular experimental procedure. And then, of course, the overall data set that it was a part of. And moving forward, more and more information about the protocols that were used there. Again, FaceBase has a diverse collection of data, and there are several different categories. I mentioned a few here. It can range from sequencing data to the process data, you know, alignment files and such, gene counts, peak files, then tracks and images, which can be microscopy. It could be, we have some histology, we have, or a little bit of histology, and then, and certainly CT and MR kind of imaging data, a small amount of surface models, and then even some, you know, older data, say would be found in microarray types. So this is the overall layout of how data and the metadata are organized in FaceBase, and should give you kind of a picture for what you're looking at when you actually get down to FaceBase data, you know, the display in the data browser.